My great pleasure today to introduce Dr. Roger Cohn, who our speaker for the drug discovery seminar series. So Roger studied at Princeton and MIT, and was on faculty at Oregon Health Sciences University before moving to Vanderbilt, where he was for 20 years?
8 years of
8 years at Vanderbilt. And then about 3 or 4 years ago, he came to the University of Michigan to run the Life Sciences Institute there, which is quite similar to our IQ Institute. It's a multidisciplinary institute where all the faculty have academic homes in other departments. Really spectacular group of faculty. And one of the big initiatives there is academic drug discovery. I was lucky to be involved with them from the beginning in setting up their screening center, and so it's been very effective and done a lot of exciting things. So Roger has done a lot of work in energy homeostasis, diabetes, GPCR control, especially the melanocortin receptors. Done some critical work in that area. He's a member of the National Academy of Sciences, National Academy of Medicine, and recently, in addition to his role as the director of the Life Sciences Institute, he was also named Associate Provost for Biomedical Sciences or Biomedical Initiative. They've got a very exciting program, something like RGII here that they've just initiated there, but specifically in biomedical sciences, which has been moving along. So welcome and thanks for coming, and we're looking forward to hearing your talk.
Thanks, Rick. So, of course, Rick played a major role in launching academic drug discovery at Michigan with the development of the Marine Corps there. So he and I have been talking for a long time, and finally got a date to come up here. So thank you very much for this work. I've spent almost my entire career focused on the melanocortin receptors. There's 5 melanocortin receptors, and I named them MC1 through MC5. Very clever names. Almost all of my work has been on the MC4 receptor, but over the last few years, I think we've finally been able to understand the MC3 receptor, and I think it's fascinating and has some very interesting applications. So let me give you a quick review of the central melanocortin system, where the MC3 and MC4 receptors are expressed. This set of neural circuits defined by, let's see, we have a pointer.
I think there's a stick, or use the.
I can use this, I suppose, yeah.
Yep.
So this central set of circuits is defined by neurons in the arcuate nucleus that express the agonists for the melanocortin receptors derived from the POMC gene, propionmelanocortin, and one of the few endogenous antagonists of a GPCR, the AGRP protein, which is made in a separate set of neurons in the arcuate. And these neurons, indicated in red and blue here, co-project to a number of centers that regulate energy homeostasis, shown in yellow, that express either the MC3 receptor or the MC4 receptor, or both. And pharmacologically, the point is that this system provides a bidirectional control of energy homeostasis. So in this experiment from Novo, they're characterizing one of their MC4 peptides, administering it here to mini pigs, and over 2 months, they show chronic induction of weight loss with no tachyphylaxis, with 0.3 mg/kg of this stabilized MSH peptide. This is a very elegant experiment from Malcolm Lowe's lab at Michigan. So this is the growth curve of a normal mouse. This is a growth curve in which the POMC gene, encoding the agonist for the MC3 and MC4 receptors, has been genetically inactivated. You can see the animal exhibits morbid obesity, which is what happens in humans with POMC mutations or MC4 mutations. And then he reactivates the POMC gene hormonally. It's inactivated, but for the hormonal reactivation. And you can see that those animals then lose about 80% of the excess weight that they put on due to the lack of the agonist. So this system, again, provides bidirectional control of energy homeostasis. The normal endogenous ligands are derived from peptides cleaved from POMC, and they come in a variety of flavors, but they all have this core pharmacophore of HISPY, ARCH, TRP, alpha, beta, gamma, MSH, and ACTH. Additionally, there is this endogenous, ultimately it becomes a 50 amino acid cysteine knot protein called AGRP, which also binds to the MC3 and MC4 receptor and blocks MSH binding, but has some other unusual activities of its own. These are some of the early compounds, synthetic analogs that have been used to derive pharmacological development of therapeutics. And they're all from Victor Ruby's lab. NDP-MSH, putting a D amino acid at this position, increases affinity and stability. And then cyclic compounds, which are stabilized by virtue of being cyclized and containing the D Phi. Increasing bulk at this position produces the antagonist. Anyway, we're now in the age of melanocortin therapeutics. Multiple compounds are already in the clinic. 2 are approved. And this one here that I'll focus on is basically just a cyclic analog of MSH called setmelanotide. And interestingly is able to reduce food intake and reduce body weight without any pressor side effect. Many of the potent small molecule compounds have pressors, target-mediated pressor side effects. Anyway, as I was indicating, 2 compounds, Vyleesi and Scenesse, are now in the clinic. 2 melanocortin are now approved. Scenesse is an MC1 agonist approved for erythropoietic protoporphyria. It basically increases tanning, which is essential for ameliorating this condition. Vyleesi was just approved for hypoactive sexual desire disorder, developed by the company Palatin. Another aspect of MC4 is it regulates sexual function. And setmelanotide is about ready to be approved for syndromic obesity. This compound has enough activity that the reproductive effects are not problematic in this compound. It produces weight loss in syndromic obesity, but is not potent enough to be useful yet for dietary obesity. So we still need additional research to try to figure out how to utilize this very, very well-validated target for dietary weight loss. But anyway, I'll show you the data for setmelanotide action in syndromic obesity. It's highly effective in POMC deficiency, very rare condition, but multiple patients worldwide. It's recently been published to be effective in leptin receptor deficiency because a good 50% or more of leptin action requires the melanocortin circuits, so it's upstream of MC4. And recently was just shown to be highly effective in Bardet-Biedl syndrome, another syndromic obesity, which is much more common than these other 2. So this company, Rhythm, is gradually trying to build a market for this drug by identifying types of syndromic obesity dependent on MC4. But anyway, here you can see normal growth curves. And this is what happens in all forms of melanocortin insufficiency, either mutations in the ligand or the receptor. From 6 months of age, kids become morbidly obese, and you can see the degree of obesity compared to the 97th percentile for a normal growth curve in kids. When this drug is administered, you can see weight loss, weight change in kilograms in red. After only 40 weeks of treatment, one patient has lost 60 kilograms already with no tachyphylaxis. You can see the hunger scores in gray. So this is a miracle cure for these kids with morbid, highly morbid early onset syndromic obesity. But as I indicated, nobody yet has been able to get adequate potency without this unwanted pressor effect. And this shows the pressor effect here. You can actually see this just by looking at mouse models where you knock out one or both copies of the MC4, where you knock out one or both copies of the MC4, you can see decreasing mean arterial blood pressure as a function of MC4 receptor dose. And then some of the compounds which were killed in clinical trials, a Lilly compound shown in green, a early Palatin compound shown in purple, produce a significant increase in blood pressure rapidly after the compound. In contrast, the Lilly compound, where I showed that weight loss in mini pigs, has no pressor effect compared to vehicle. Nobody's figured out whether this is biosignaling, pharmacokinetics, so lots of pharmacological mysteries. And one of those mysteries is the MC3 receptor. When we first cloned the MC3 receptor back in the day and mapped its expression along with that of the MC4 receptor in the brain, we noted that they're expressed in very different places in the brain. Importantly, in the very POMC and AGRP neurons in the arcuate nucleus, we noted that only MC3 is expressed. And then MC3 is also in critical behavioral sites, like the ventral tegmental area involved in reward, the paraventricular nucleus of the hypothalamus involved in anxiety, for example, and many nuclei where you only see one receptor or the other, and then there's some nuclei where you see both MC3 and MC4. So, we immediately knew from the neuroanatomy that these receptors have some very different roles. Early on, Michael Cowley in my lab hypothesized, I wouldn't say proved, but hypothesized in this paper that MC3 was acting presynaptically on AGRP neurons to suppress POMC neurons, inducing release of GABA onto POMC neurons. The reason I say did not prove is because pharmacologically he used a compound with really limited MC3 specificity to make this hypothesis. But in any event, the neuroanatomical data had been around for some time from Tomasz Horvath and others showing that NPY-AGRP neurons project and synapse onto POMC neurons, but not the other way around. So this is clearly, even though these anorexigenic POMC neurons and orexigenic AGRP neurons are both in the arcuate, they form a unit that's communicating internally. Well, one of the reasons why we and almost nobody else worked on the MC3 receptor is that the early biology that came out of our initial knockout studies was really confusing. In most obesity models, when you increase fat mass, you also see an increase in lean mass. This is true in human dietary obesity as well as all the different syndromic forms. So when we knocked out these receptors, we saw morbid obesity, 48 grams in the mouse compared to a normal weight of 26 in that same background strain. And we saw a really mild sort of later onset obesity in the MC3. Furthermore, while there was a very measurable increase in lean mass in the MC4 obesity model, there was actually a significant loss of lean mass in the MC3 knockout. And so that didn't make any sense really. And then furthermore, this modest late onset obesity in the MC3 knockout model did not produce any measurable effect. There was no measurable effect on food intake or energy expenditure when the animals were just fed ad lib on normal chow. That mild obesity was also contradicted by two other findings. The MC3 knockout mouse was hypersensitive to cachexogenic stimuli, like tumor implantation or IL-1 treatment. So in some cases, the receptor seemed to be acting in an anorexigenic way, thus knockout producing obesity. In some cases, the opposite. The receptor seemed to be constraining reduced food intake and weight loss. And then when we use this compound D-tryptase gamma MSH, which has about 50-fold selectivity of MC3 over MC4, at the right dose, we were actually able to see stimulation of food intake. And we knew it was MC3 mediated because we didn't see it in the MC3 knockout animal, at least it required the MC3 receptor. So anyway, we were getting all these contradictory findings. Sometimes the MC3 seems to stimulate food intake, sometimes it seems to be involved in inhibition of food intake. And so in the light of all this contradicting data on the role of the MC3 in energy homeostasis, we really didn't get any kind of systematic understanding of what this receptor did. This just shows you some of our data on cachexia. The MC4 receptor shown in the bottom two panels blocks cachexia, tumor-induced cachexia. So if you look at lean body mass, for example, in a sham animal, you get increase in lean body mass as the mice grow, but when you implant a tumor, the MC4 in the tumor-containing animal, the MC4 actually restores some of that increase in lean mass that's not seen in the wild-type tumor implantation model. In contrast, in the MC3 knockout animal, with the tumor implantation, we saw an increased loss of lean mass in this cachexia model and increased loss in obese mass as well compared to MC4, which blocks cachexia. So enhanced tumor cachexia in the absence of the MC3. So this just shows you, the claim that MC3 does not affect normal food intake. This is just normal food intake in wild-type MC3 and MC4 knockout animals, the work of Masoud Gemar-Langrudi in my lab at Vanderbilt. And even in young, lean animals that are matched for lean mass, you can show hyperphagia when you knock out MC4. You don't see any effect of MC3. What we had noticed early on, though, was that there was a defective fast-induced refeeding in the MC3 knockout. It's very common when you study food intake to fast animals overnight to make them hungry, and then look at the way the genes or drugs affect the refeeding. And we had done that model, and we noticed that whereas there was normal refeeding in the MC4, the MC3s were defective in fast-induced refeeding. This effect was pretty profound, too, because a single overnight fast produces 4 days of defective daily food intake. So they're hypersensitive to fasting, and they don't start eating normally for several days after a single overnight fast. If you look at this in terms of cumulative food intake, cumulative food intake is not normalized for a full 10 days after a single overnight fast. Well, it turns out this was more than, so when we think about energy homeostasis, we say there are 3 effector pathways by which animals regulate energy homeostasis: behavioral, endocrine, and autonomic. So I showed you behavioral defects in response to a fast. We also discovered that there were neuroendocrine defects in response to a fast in the MC3 knockout animal. So for example, they have a normal HPA axis response to stress, MC3 knockouts, when you measure serum corticosterone, but they exhibit a defective HPA reduction in response to a fasting stress. And furthermore, we saw defects in elevation of AGRP and NPY mRNA. We went on, and some of this is unpublished, we went on and measured other neuroendocrine axes to see if likewise there were defects in response to fasting. And this is classic when you fast a mouse, or for people who are anorexic or not having adequate energy stores, you will see suppression of the thyroid axis. And this can be demonstrated with an overnight fast in a mouse. An overnight fast in a mouse, as many of you probably know, is like starvation. I mean, they lose a lot of fat and they exhibit a full neuroendocrine response. And that's shown here, looking at total T4 hormone in a wild-type animal in red, and then after fasting in purple, you can get a measurable decrease in thyroid hormone with just an overnight fast in a mouse. In contrast, in the absence of MC3, there's no measurable decrease in thyroid hormone. Similarly, when you fast a mouse overnight, you will prevent reproductive cycling for up to a couple weeks. A simple overnight fast will do that in a mouse. In contrast, in the MC3 knockout, that animal continues cycling reproductively between estrus and diestrus normally, even after an overnight fast. And I won't show you the control data here, but suffice it to say, the MC3 knockout animals lose the same amount of weight as the wild type, and they have the same decremin and leptin, which controls all these things through the melanocortin and other circuits. So it's not due to a defect in weight loss or fasting-induced decremin and leptin. So this is unpublished data now. Ultimately, we believe this is due to a defect in leptin-mediated regulation of AGRP neurons following fasting. So normally when you fast a mouse, leptin goes down. And due to leptin receptor expression in these neurons, as well as glutamatergic input into these arcuate AGRP neurons, you get a very strong upregulation of the AGRP neurons. And that's shown here by CFOS, and here are the images. When we quantitate this, you can see a several-fold upregulation of CFOS in the arcuate in wild-type animals. In the MC3 knockout, that's blunted about 50%. And this just gets to the weight loss. Here you can see comparable weight loss. Comparable weight loss in both the wild-type and MC3 knockout animals when we fast them. So this is one effect of MC3. It's required for the normal sensing of nutritional state by the AGRP neurons. This is not a developmental effect because we can mimic all of this with acute suppression or knockdown of MC3. So here we're using lentivirus injection into the arcuate nucleus. This is the behavioral assay looking at food intake, and you can see very similar magnitude and kinetics. We disrupt daily 24-hour feeding after a single overnight fast in these lentiviral suppressed animals, and we disrupt cumulative food intake just by suppressing MC3 in the arcuate, knocking down MC3 in the arcuate. So the conclusion is that MC3 is required for normal behavioral and neuroendocrine responses to an orexigenic challenge, a challenge to stimulate food intake and overnight fast. And here is the rather amusing consequences then. I just showed you orexigenic stimuli and overnight fast, but if you administer anorexigenic stimuli, you get similar responses. And so what if you play this out over long term by both dieting an animal? Well, in the MC3 knockout, like I showed you with the overnight fast, if you have chronic food restriction, so you give them 80% of what they would normally eat ad lib, they lose more weight than wild-type animals. They don't come back to their homeostatic set point like the wild-type animals do. Here's their original weight. The wild-type animals, when you put them back on chow ad lib during this time period, they come back to their previous weight and plus a little bit. The MC3 knockouts come back in and come into a set point below their previous weight. Then after ad lib feeding, if you switch them to high-fat chow, the MC3 knockout animals gain more weight. So in contrast to MC4 and leptin mutations in mice and people, with MC4 or leptin mutations in mice and people, what happens is the animals cannot establish a set point. They simply continue to add more fat and become morbidly obese without achieving a set point at all. In contrast, MC3 knockout animals exhibit this behavior. When there are things that are driving weight loss, they lose too much weight. When there are stimuli that are driving weight gain, they gain too much weight. And so we hypothesized, therefore, that MC3 is required for energy rheostasis, the regulated movement of set point. In other words, if you're going to move set point, you have to impact these boundary conditions. Any homeostatic system involves a set point and boundary conditions. When you approach the upper boundary, you launch effector pathways that bring you down. When you approach a lower boundary, there have to be effector pathways that bring you back up to this set point. And so you bounce in between these two boundary conditions. And our argument, our hypothesis is that MC3 is required for the regulation of these boundary conditions. And that's why when you do nothing to an animal, you don't really see anything in the MC3 knockout animal. You have to perturb them one way or the other. So we believe MC3, and this is a term that's not been used in the energy homeostasis field, rheostasis. But there are many conditions in which you change your set point. Pregnancy, in many mammals, hibernation, disease, aging, change your set point. So to further address the mechanisms by which MC3 might be regulating rheostasis, we turn to our electrophysiological model where we focus in on the PVN neurons expressing MC4, because these neurons are a critical site of regulation of feeding behavior by MC4. So over the years, Masoud Gamar-Langrudi in my lab developed very nice eFIS slice preparations where he could record from MC4 containing neurons to look at their behavior. And so I'm just showing you this data. I'm not going to go into the coupling to the science, but I just want to show you this figure because it shows you what a nice system this electrophysiological slice prep is. So when we take this preparation and patch onto these MC4 neurons that have been identified under the scope by virtue of expressing GFP under the control of MC4 promoter, MSH increases firing frequency and depolarizes these cells. So we can just put MSH in the dish and 90% of the neurons will show an increase in firing frequency and a decrease in membrane potential. And interestingly, these neurons retain the AGRP effect. So when you put AGRP in the dish, most neurons will show a decrease in firing and an increase in a hyperpolarization of neurons. So it's a great model system for characterizing melanocortin effects in a native cell expressing the MC4 receptor. Now, in this preparation, Masoud had noticed for years and really hadn't done anything with the fact that he could measure MSH-regulated inhibitory currents as well. And so typically when under the right conditions, when he put MSH in the dish, he could also see an increase in inhibitory currents in these PVN MC4 neurons. And so to understand the pharmacology of this, he repeated these experiments, recording from MC4 neurons in a background in which either the MC3 or the MC4 receptors had been knocked out. And what he found was that knocking out the MC4 receptor did nothing. GABA currents were still present, but when he knocked out the MC3 receptor, the GABA currents were gone. And so what he had identified was an MC3 mediated regulation of GABA release onto these PVN MC4 neurons. Well, it turns out the bulk of these neurons are indeed the AGRP neurons, because when he uses cholera toxin to kill the AGRP neurons, he gradually sees a diminution or complete loss, actually by day 7 or 8, of the MSH-induced release. So here you see at day 1 after cholera toxin administration, you can still see MSH-induced increase in inhibitory currents. But as you kill off the AGRP neurons, those go away. So he's recording from, of course, from the PVN, but looking at the effect of the AGRP neurons on this. And so the model that we put together in this paper that we published last year is shown here. It's been known for a long time that the anorexigenic POMC neurons release MSH onto MC4 neurons all over the brain, and that AGRP projections are found in almost all of these sites, also projecting to MC4 neurons. The microanatomy is not really understood yet. How do you have neurons releasing an agonist, an antagonist, inverse agonist, regulate the same receptor sites? Is it volume release, et cetera? There's only been one paper on that, and so we really don't know the understanding of the microcircuitry. So this drawing is a little bit of fiction here in terms of the geography of all of this. But in any event, pharmacologically and electrophysiologically, we know that POMC neurons release MSH onto MC4 neurons, which activates these neurons and inhibits food intake. And we know that that MSH, when it acts as MC3 neurons, dumps GABA onto these neurons. So you may have a complete regulatory circuit here where these neurons are anorexigenic and ultimately enough MSH spills out to produce a counter-regulatory release of GABA onto the very same neurons. So you can imagine if you remove this input, you're going to have unrestrained anorexigenic drive. If you hyperactivate this unit, you're going to stimulate food intake. And so the finding, so Stan Watson actually at Michigan decades ago published a paper showing that 50% of POMC neurons and 50% of AGRP neurons express MC3 by C2 hybridization. So this raised the possibility for us that MC3 agonists may be used to regulate AGRP neurons. And this could have a number of interesting effects. First of all, there's a lot of optogenetics and chemogenetics out there on the AGRP circuits. These are the most profoundly orexigenic circuits known. And if I can get the video to play here. When you use chemogenetics or optogenetics, in this case it's chemo, to activate the AGRP neurons, you can literally just walk in the animal room and immediately tell which animals have the activation of AGRP neurons. The wild type, the AGRP animals, this is first thing in the day after they fed ad lib all night. They're just hanging out at the food, eating food nonstop. Whereas the wild type animals are sitting in the corner, not eating and sometimes sleeping. All the AGRP animals are at the hopper eating. So activation of AGRP neurons produces profound stimulation of food intake, even in animals that are fully sated. So the first experiment we thought we would do would be to chemogenetically turn on just the MC3 neurons in the arcuate nucleus, taking an MC3 Cree animal and injecting inhibitory or excitatory dreads into just the arcuate and ask, well, can we just access the arcuate? Because the arcuate's very druggable. It's right next to the median eminence, and a lot of peripheral hormones like leptin act on the arcuate. So we know it's a really good druggable site. So we thought we'd just test the MC3 arcuate neurons. Now, one of the reasons why this is not a trivial experiment, I told you that already that the POMC and AGRP neurons both express MC3. So you could get a washout where you're both stimulating and inhibiting food intake and nothing happens. And so in collaboration with Rich Simmerly, we've been doing detailed neuroanatomy of MC3 all over the brain. And in the arcuate, the triple fish experiment is shown here, which is using in situ hybridization to identify MC3, AGRP, and POMC expression. The AGRP and POMC neuropeptides are expressed at such high levels that it basically just looks like immunohistochemistry almost. And then you can see lots of, each of these dots is probably an individual MC3 mRNA molecule in an AGRP neuron and a POMC neuron. And we got 97% of AGRP neurons making MC3 by this more sensitive method, and about 80% of POMC neurons. So it was not a trivial experiment to ask if we activate both of these cells by using dreads, what happens? Also, let me point out, there's a very large number of non-POMC, non-AGRP neurons in the arcuate that express MC3 as well. We have not identified them yet. In any event, we got a very striking result, and by we, I'm talking about Patrick Sweeney, who's done all of the MC3 work I show you here today, an amazing postdoc in my lab. So when he activates MC3 arcuate neurons with a dread, he gets profound stimulation of food intake. This is a single treatment with CNO. And he gets 6% body weight gain in one day after one injection of CNO. So profound stimulation of food intake and weight gain. When he inhibits MC3 arcuate neurons, he gets inhibition of food intake and chronic weight loss. So 5 days of weight loss when he gives CNO chronically here. And so this was really striking, suggesting that there is a unit of neurons here that can be singularly regulated via MC3. And so again, this is just to point out that we're just looking at this one little bit of melanocortin receptor expression here in the arcuate. Importantly, there's beautiful data from a number of laboratories. Michael Crash is at the NIH, Brad Lowell, Zach Knight, and others, showing that the AGRP circuitry does more than just sense hunger. So, of course, the arcuate is a major site of sensing of leptin levels. So when you diet and exercise and lose weight, a decrease in leptin is activating AGRP neurons in the arcuate. But more than sensing energy and hunger and driving food intake through regulating centers like the PVN and the VTA, Knight and Lowell and others have shown that additionally the AGRP circuits suppress a number of other motivational states. So they suppress anxiety and stress, they suppress alarm via projections to the PBN, they suppress fear via projections to the amygdala, and you can do experiments like paint fox urine on the food, and normally mice will stay away from fox urine no matter what. But if you fast them and look at their interest in eating, they will not do so. If you stimulate the AGRP neurons, they will even go into the corner where the fox urine is consumed. So you're suppressing. So these in order to allow feeding to take place, these neurons also suppress other motivational behavioral states. So we've now tested that in a number of different MC3 models using a variety of behavioral assays where the animals are either sated or fasted. So with animals fed ad lib using the elevated plus maze, when we activate MC3 neurons, they'll spend more time in the open arms, indicating reduced anxiety. When we suppress MC3 neurons, they'll stay out of the open arms, indicating an anxiogenic state. This is called the novelty suppressed feeding assay, and this is important, as I'll show you in a moment. So here what we do is we fast the animals overnight and put a piece of food in the middle of a large open cage. If you look at the videogram here, you can see that normal animals will tend to huddle in the corner. They don't want to go into the middle, even if they've been fasted, because that's a scary place to be. They could experience predation. If we activate the MC3 neurons, they spend less time in the corner and more time in the center. And that's quantitative here. It doesn't affect locomotion. So distance traveled is the same. They still wander around in the corners, even when you activate the MC3 neurons. So we're not just impacting locomotion. In contrast, when we inhibit the MC3 neurons with a dread, we're creating an anxiety, we're increasing anxiety, and they huddle in the corners. So this circuitry regulates anxiety, whether they're sated or fasted. And in this case, you're looking at the competition between hunger drive and the fear of going into the center of an open cage. We've done this in the MC3 knockout model as well, and we see the same thing, again, either using the open plus maze diagrammed here, where the mouse wanders around and you videograph the time it spends in the different parts of the device, or this is the open field device that we use, again, quantitate the video images. And when you knock out the MC3 receptor, they're more anxious and they spend less time in the center or in the open arms. So all of the data I've shown you has been reproduced in male and female mice. And in the knockout, however, with the dreads, let me back up. With the dreads, when we do these experiments, just looking at the arcuate now, in male and female animals, we see no difference in gender. However, when we went back to the knockout, and this is a global knockout now, MC3 throughout the brain, we got a profound sexual dimorphism. So now we're running again the novelty suppressed feeding assay. And the males, this is with the knockout, not the dreads, the males in the knockout, in the case of the knockout, really don't show any difference in terms of latency to go into the center to explore the food. In contrast, the females have a significantly increased latency. They don't like going into the center, and they spend less time in the center compared to the males, where we saw a small difference, but it was not significant. Furthermore, if we actually measure food intake in this model, where we're introducing two stressors, we're fasting the animal overnight, and we're making them go into the center of an open field to consume food. Only 7% of the males don't eat. 80% of the females were aphagic. They sat in the corners and never ate. This is, I think, really important because this looks like anorexia nervosa in the sense that there's a very large sexual dimorphism in anorexia nervosa as well, with 95% of cases, 90 to 95% of cases being in women. And it's been argued, particularly by clinicians, that there are no good models of anorexia nervosa. And one of the reasons why is there is models that are out there, like the activity-based anorexia model, don't show any sexual dimorphism. So here we found a profound sexual dimorphism in anorexia. So we're very excited about this because we think this is a very druggable region of the brain accessible to peptides. This shows the MC3 neurons in green using GFP and their fiber projections from these very same neurons using immunohistochemistry for AGRP. And you can see some of these projections even go down into the median eminence, which is fully outside the blood-brain barrier. But the arcuate has a lot of properties of a circumventricular organ, and we know it directly senses a lot of peripheral hormones and peptides like leptin. So anorexia nervosa is a really serious disease. I'm going to tell you a little bit about it. It's characterized by a persistent restriction of energy intake and a number of neuropsychiatric conditions, including intense fear of gaining weight, a disturbance in self-evaluation of body weight. So for example, typical restricting type anorexia nervosa patients will say they're fat, even though they're well below a healthy body weight. A very stereotypical peripubertal onset and a number of comorbid neuropsychiatric conditions, including depression, anxiety, and OCD. About, as I said, 90% of cases occur in women. It has a very high prevalence. The epidemiological data suggests a prevalence of somewhere between 1 to 4%. And very few people know this. It's got the highest mortality rate of all of the psychiatric disorders. Something like 18 to 20% of people with restricting type anorexia nervosa will die of the condition. There is currently no effective pharmacological option, and drugs used for depression and anxiety don't work in anorexia nervosa. Nobody understands why. So SSRIs are ineffective, for example, and the current modality is family and individual psychotherapy. Here's what's really striking and gets to the NIH issue here. Whereas the NIH last year funded 2,350 grants on depression for a total of 252 million, 1,300 grants on schizophrenia for 180 million. Last year they funded 45 grants in anorexia nervosa for a total funding of $5 million. We decided to focus on the MC3 as a potential target for anorexia nervosa because it's an eminently druggable receptor in a region of the brain we know we can access. And it not only stimulates food intake, but most importantly, seems to inhibit other motivational states at the same time, which could be really useful. So we've identified some MC3 specific peptides that we can use, and the early preliminary data that we have on feeding is outstanding. And this is all Patrick's data shown here. Double 6-hour food intake gives us a 20-25% increase. This is, again, in the mouse model, we get about a 20-25% increase in 24-hour food intake, which is a critical hallmark in animal studies in feeding behavior. Oftentimes, you can inhibit acutely and get satiety, but then animals will make up for that during the rest of the circadian cycle. We get stimulation of 24-hour food intake, and that stimulation is completely gone in the MC3 knockout animal. So again, about 4 grams per day of food intake, but no effect in the MC3 knockout background of this MC3 agonist peptide. Furthermore, if we inhibit the arcuate MC3 neurons with CNO using the DREAD model, here's the non-CNO treated animals, the same dreaded animals but without CNO treatment, MC3 giving nice stimulation of 4-hour food intake, no effect in the CNO treated animals where we've suppressed the AGRP neurons in the arcuate. So it looks like it is the AGRP circuits, it is MC3 mediated via the arcuate. And Patrick just got, let's see if I can get this video going. He just got endomicroscopy working in the PVN MC4 neurons. So we're using a GCamp to label the MC4 neurons. This is spontaneous firing of these neurons in real time. Here's the lens implanted just above the PVN. And then when we quantitate the activity of MC4 neurons, we can show that an MC4 agonist administered peripherally increases the firing of these neurons in real time in live behaving animals. And this is another MC3 antagonist. Given peripherally, we can inhibit the firing of MC4 neurons in the PVN with administration of this MC3 agonist compound. So we've got a lot of nice tools to study the kinetics and the pharmacology and receptor occupancy, if you will, or a proxy for receptor occupancy. So we're very excited about pursuing MC3 agonist peptides for eating disorders like anorexia. I showed you a lot of data on inhibition of food intake too, though, when we inhibit the MC3 receptor or knock out the MC3 receptor. So interestingly, we think this is an interesting target to pursue for obesity as well, although we have to address the critical issue of whether MC3 antagonism will also induce a suppressor effect. Nonetheless, in early experiments, the MC3 antagonist and all of these compounds, the agonist and antagonist, have over 1,000-fold specificity for MC3 over MC4. Here we show an acute inhibition of feeding assay with an MC3 antagonist. We get about 75% inhibition of feeding in a 6-hour assay, and we get, again, 25% inhibition of feeding in a 24-hour food intake assay. And that effect on food intake is absent in the MC3 knockout. So we believe we're really addressing the target here. Well, probably the leading drug on the market right now for dietary obesity is one of the GLP-1 compounds, liraglutide. This shows change in body weight, 24-hour change in body weight in a mouse after administration of an MC3 antagonist, liraglutide, or the two drugs together. So inhibition of MC3 also synergizes with liraglutide activity. And we haven't looked at the metabolic consequences of this yet, but it'll be very interesting to pursue the mechanism to see how this enhances liraglutide action. The famous Detroit industry murals from the DIA, which some of you I'm sure recognize. I want to just briefly touch back on this sexual dimorphism, because we've got a neuroanatomical basis for it, and we're going to work on trying to figure out exactly what the critical circuits are. We actually published some data on this a few years ago. Rachel Lippert, a graduate student in my lab, showed that MC3, but not MC4, was expressed in the VTA, and about half of it is co-localized with TH. And when you look at dopamine content in the VTA, in wild-type male animals, there was no effect of deletion of MC3 on dopamine content in the VTA. But when we knock out MC3 globally in animals, you can see a significant increase in dopamine in the VTA in these animals. And we see some small effects on sucrose preference as well. I won't go over that. So in sham OVX animals, you can, as I show you here, measure that increase in dopamine. When you OVX the animals, you normalize them. So it is a reproductive phenotype, this difference.
I'm confused by this because the MC3 is in the VTA neurons, TH positive neurons.
Correct.
It stimulates cyclic AMP.
It's also in non-TH neurons, and this is the global knockout. So honestly, we don't know. That's a good question.
So I would have predicted that the knockout, if anything, would have lower dopamine, not too much dopamine.
Yeah. Yeah, we don't know whether this is from GABAergic MC3 neurons coming from somewhere else or specifically.
Yes, if you could do a conditional in the TH positive neurons, then you could get at that more directly. Yep.
We have no idea what the circuitry is that's underlying this effect. But just the point is to say there's some profound sexual dimorphisms linked to MC3. And actually, I observed this in human hypothalamus before we did any of this work. I was just noodling around in the GTEx sample set, and all the melanocortin genes, the POMC gene, MC4 gene, there's no sexual dimorphism of expression. This is transcriptomics from the GTEx sample set in humans looking at hypothalamic MC3. This is a log scale. There's about 5 times more MC3 mRNA in human hypothalamus in males, in male human hypothalamus versus females. So once I saw that, I got really super intrigued. And Patrick then showed that there is about only 50% MC3 mRNA in the arcuate in female mice compared to male mice. And Rich Simmerly has confirmed this for us by fish. You know, I showed you all those experiments talking about the MC3 neurons in the arcuate. The dimorphism is not in the MC3 neurons. It's not in the POMC or AGRP neurons. And so all the drug effects that we're seeing on food intake and the dread effects, there's no sexual dimorphism. It turns out the sexually dimorphic expression is in one of the non-POMC, non-AGRP neurons in the arcuate. We don't know which one it is yet. We'll need to figure that out. We also see the opposite dimorphic effect in kisspeptin neurons, in the anteroventral periventricular area where kisspeptin neurons are expressed. There's a lot more MC3 and kisspeptin neurons in female mice than male mice. We've already seen reproductive phenotypes and published them. So for example, the MC3 receptor is required for the hyperphagia and weight gain in pregnancy in mice, required for that hyperphagia. So lots of interesting work to do. And again, you know, I pointed out that we're doing drug discovery. We're focused in on both the arcuate and peptide drug discovery because we think that's a very druggable region, and we've shown you from these dread experiments that we can get profound phenotypes just from regulating the arcuate MC3 neurons. But we're also interested in small molecules because MC3 is in all these behavioral regions. Richard Palmer has told me there's more MC3 in the parabrachial nucleus than there is MC4. Parabrachial nucleus seems to be involved in, maybe involved in PTSD. So there may be behavioral uses for MC3 compounds if you can get into some of these deep brain regions regulating behavior. But in any event, we've got high throughput screens going for MC3 agonists and MC3 antagonists at the screening site at Michigan. So interestingly, when we did our first pilot screen for antagonists, almost all the compounds that showed better activity at the MC3 receptor than a control beta adrenergic receptor counter screen were natural products, which is good and bad. So we actually found, for example, that polymyxin is a full antagonist of the MC3 receptor shown here. Not a good starting point. It's got toxicity and this and that, but just the point that that you can find some interesting natural products that may be good starting points for MC3 and MC4 antagonists. And in data that I'm not going to show you, we know that the binding pockets very kind of spread out and shallow and may require something like a natural product. Anyway, to wrap up here, our terminology that we're using is that the MC3 regulates energy rheostasis. It's required for controlling the boundary conditions for energy homeostasis. And our data indicates it's regulating both the upper and lower boundary. So clearly I've shown you a couple of mechanisms whereby animals are going to lose too much weight and we're not regulating that lower boundary properly. The AGRP neurons don't activate properly when leptin levels get too low and animals lose too much weight. If you lose the MC3 receptor expressed presynaptically on AGRP fibers, you're not going to induce GABA release to stimulate food intake properly. So those are two molecular mechanisms whereby MC3 could regulate the lower boundary condition of energy homeostasis. I don't really have a hypothesis yet for how MC3 is regulating the upper boundary, but in at least 3 different conditions we've shown animals gain too much weight when they lack the MC3 receptor. Again, one mechanism is regulation of GABA release via these presynaptic MC3 sites on AGRP neurons. And another mechanism being the requirement for MC3 in terms of regulation of AGRP responsiveness to leptin and fasting state. I've shown you some preliminary data arguing that MC3 agonist administration phenocopies AGRP activation. You can get this profound stimulation of food intake with MC3 compounds, even given peripherally. And I've shown you our data that MC3 expression is sexually dimorphic in both mice and people. And can show you the data. Some of it's published. It's required for normal regulation of energy homeostasis by reproductive state. So we're highly focused now on the development of MC3 peptide and small molecule compounds as potential therapeutics. And sorry, I had to slip in one little football analogy here, the famous Schembechler quote about the team. And so this is the team, really Masoud Gamari Langrudi launched us into this whole discovery program on MC3 when he discovered that MC3 acts presynaptically on HRP neurons to release GABA. And he remained behind, unfortunately, at Vanderbilt. I couldn't pry him out of Vanderbilt and Nashville, but Patrick Sweeney has done all of the physiology, the electrophysiology, the dread work on the MC3 receptor that I showed here today. An amazing postdoc group. Luis Jimenez has done all the drug discovery at MC3. He's a fantastic pharmacologist, was in Sevagrovich's lab for a while, and other folks in the lab working on other aspects and other problems. So happy to take any questions.
Since the effects, thinking pharmacologically, since the effects may drive weight one way or the other, certainly if you're thinking, I see the story for the anorexia. If you were using this, say, for obesity or weight gain, you could potentially drive the weight down with an antagonist, but then if someone binges, they're just going to go crazy. So you're going to end up with huge fluctuations in both directions. So
I mean, so it's interesting in that regard. And furthermore, the mechanism is different from any other obesity drug on the market or that most people think about. It seems to be regulating food intake, but more importantly, it's regulating the compensatory or the homeostatic responses to weight loss, blocking hunger, blocking neuroendocrine response to get you back to weight. So it may be the kind of drug that works great if you're losing weight through diet and exercise, or if you're on liraglutide, for example. But if you're a mere mortal and you tend to binge, and it could be problematic.
And would combination with MC4 ligands be beneficial based on the neurocircuitry?
Well, the problem with the MC4 ligands, of course, is that when you have a potent orthosteric agonist acting at all the MC4 receptors, you get the pressor response. It is target mediated. We don't know the answer to this yet. It is possible that we can avoid entirely that pressor response with MC3 agonists. Why? The AGRP neurons don't project to a lot of the brainstem sites. There's a ton of MC4 in the brainstem, and not a lot of projections of the AGRP neurons to all the brainstem sites. So there's a possibility we'll avoid all the pressor effect with an MC3 agonist.
It's the expression of the molecule regulated by circadian rhythm.
There's probably something on the literature. I have not done that. I don't know the answer. That's a very good question. I mean, I would love to look in like hibernation models, for example, to see if MC3 is involved in allowing animals to change their set point, because animals do that. They eat voraciously in the fall, and then they just quit eating when they go into hibernation. So could there be a role for MC3 in that?
I was actually thinking also about the variations in weight of people, right? Just
right.
When you sleep enough, or you have weird patterns.
Yeah. No, we haven't looked. That's a very good question. Yeah, we should.
So this very long-term effect of fasting, there's one overnight fast in the mice, and you get an effect that lasts, 10 days?
Yeah.
Any idea what the mechanism of that is?
No, but we've got some good model systems to look at that, so we can go in and quantitate things in the AGRP neuron and see, in the absence of MC3, when you fast them, what do you break, and why does it take 10 days to come back to normal? So we're, this was one of the aims of our grant, which may or may not get funded depending on the government. So I'm hanging out there waiting for the no right now. And yeah, one of the aims is to, pull out the AGRP transcriptome and test the obvious candidate hypotheses, like maybe the MC3 is required for a transcriptional profile, and you don't get leptin receptor in AGRP neurons in the absence of MC3. So we're going to look at the obvious candidates and signaling pathways that could produce a defect to leptin response. And then we can do the kinetics on those genes and whatnot to find out the kinetics. But it is fascinating.
Has the phenomenon been shown in other species besides the mouse?
Which phenomena?
The senses that by the over the fasting induced change in food recovery. That's what you were showing us, right?
Right, right.
Has that been shown in animals other than mouse?
Yes, some of the elements of what I've shown you are known in the rat. But I would be very interested in working with anybody here in what we call a large animal model, that is anything larger than a mouse or a rat, to see if so, you know, the HRP circuitry is highly conserved. We mapped it in non-human primates. It's highly conserved. So there's a good possibility that, and the pharmacology, you know, I mean, the melanocortin system has been fantastic in terms of us, you know, small animal folks and working in the mouse model, because virtually every phenotype we've characterized in the mouse has been shown in the patients. So it is highly conserved, and we suspect the same is true for MC3 and AGRP in general. But
I was very curious about your results with LBX mice. And my question is going to be a difference in women pre-menopausal and post-menopausal?
Well, again, it depends, you know, difference in what. So whereas the global MC3 circuitry is highly sexually dimorphic, the DREAD experiments and the pharmacological experiments have shown no difference at all in male and female mice. It's only when we look at specific things such as global MC3 knockout where we see those. So we're trying to map the circuitry now that's responsible for the sexual dimorphism. So as Rick pointed out, you know, we have no idea what's causing the difference in dopamine in the VTA and the MC3 knockouts in females versus males. It may be specific to the VTA, it may be some other MC3 containing circuit or even secondary circuit projecting to the VTA. So it's complicated, and it was another part of our grant to thoroughly map this circuitry. Nobody's ever mapped it. We don't know the identity of MC3 cell bodies in the VMH or many other sites. We don't know where they project to, who projects to them, et cetera, et cetera. But, you know, the data coming out on the AGRP circuitry, which is a big subset, or an important subset of MC3 circuitry, is fascinating. You know, originally everybody argued that the hypothalamic AGRP circuitry was purely homeostatic in nature. It only drove homeostatic feeding and didn't really impact behavior. And that's absolutely not the case. For example, really elegant studies from Mark Anderman at Harvard has shown that even cortical sites involved in sensory aspects of feeding behavior are regulated by AGRP. It's just indirect. So AGRP projects to the VTA, VTA projects to the insula. AGRP neurons directly regulate the saliency of visual food cues. So you can look at interactivity as a function of showing mice food, and you can regulate that response by regulating AGRP.
Just quick question. Sorry, quick question. Is there any read on whether or not you'll get a response just from pulsing the drug? Or is it going to require continuous exposure?
Well, all of our experiments have been single acute injections. That's
Right.
the best we can do in terms of pulsing. I mean, these are peptides, and some of the cyclic peptides are pretty long lasting. You know, a single dose of CNO gives us profound effects. Those were all single injections of CNO, you know, giving us a 6% increase in body weight. But yeah, you know, the kinetics are really weird and interesting, and that's definitely going to be an interesting thing to try to understand.
Well, I'd like to thank Roger for a great